other one where it's a lot harder to see. But let's start. I'm so thrilled that we're here. Where were we supposed to be today? Chicago. So we should have been in Chicago. I'm sure it's a beautiful day. There's a nice cool breeze off the river, or the lake, excuse me. And but we're here this way, and we're hoping we'll be in person at our next meeting, which is actually in Seattle. I don't know if people noticed that, but haven't been there in many years. So I'm Roy Herbst, and on behalf of this august crew, we're going to give you updates on LungMap. We're going to talk about accomplishments, update manuscripts, translational medicine, and we want everyone's input on that. We're going to talk about drug solution update, and then some of our sub-studies, one that's ongoing and another that's about to start. And then we'll hear from our site coordinators, which is so important. And then we'll have a bit about the future. So if we can have the first slide, please. And remember, there will be Q&A at the end. So welcome, and I'm Roy Herbst. I'm a LungMap study chair. I'm a medical oncologist. I'm here in my office now at Yale University, and actually just left the clinic. Next slide. We are a team, and David Gundera, is it 7 or 8 years ago you made this slide? And if you ever need a really great slide idea, the creativity master is right there, David Gundera. And what this shows is the circle of all the groups that really work together, whether it be, where we've got close collaboration with the NCI. There are some people from the NCI on SWOG, and all the cooperative groups, by the way, FNIH, the Friends of Cancer Research, a lot of consultation with the FDA. But then you can see the groups all in the circle. And then, of course, the companies. This is what we call a public-private partnership. We have the private funds coming into the FNIH and helping our public endeavor. And one of the things that I'm most proud about LungMap is that we're getting it out to so many sites. Next slide, please. If I have another slide. You can see that we're all over the country. And this is not a COVID map. This is LungMap sites, and we're diverse. You'll see in some of the presentations that we have minority accrual that's much higher than the national average. We're getting out to the populations that need these drugs, that need profiling. You can see that if you look at as of 10/8, just a couple of days ago, a week ago, you can see that total we have 4,000. We had 4,000 registrations to this trial. About 2,000 screened at progressive disease, and more than 2,000 pre-screened, meaning they're screened before they get to the refractory setting. And then you can see sub-study assignments, over 2,000, 1,647 among screened at PD, and 745 among the pre-screened. And you can see 995 patients on a drug. And you're going to see today some of the data from that. You're going to hear from David Cousino about some of the tissue we have from that. And I'd love people's ideas on how to use that. But it's a team effort. We're getting drugs to the patient where they are in a personalized way. Next slide, please. And here's our current LungMap schema. And for those who have been with us since the beginning, we started as S1400. It was a SWOG trial. Now it's LungMap. It's not just refractory squamous, but it's all refractory lung cancer. We have two major arms of this. We have the biomarker-driven sub-studies and the non-matched sub-studies. The biomarker-driven sub-studies, those in the dark blue, those are all completed. You'll hear from Mary, most of them presented and published. In fact, almost all. You'll hear about in the green, the KRAS 1900E, Suki, who now is an official SWOG member, but Suki started this working with the ECOG, so it really is a trial between groups. Will tell us about Amgen 510. Couldn't be more timely. We have another trial that's in preparation, 1900E, selpercatinib, the RET inhibitor with chemotherapy. And then we have these non-matched sub-studies. Not everyone's going to have a driver, as many as we have. And there we have the blue ones that are closed or completed. You'll hear a bit about these from Mary. But then we have one that's about to start any day now. And Hatem is on the phone. He and John Rangel, who's, I think, with his new baby today. But they have another baby in a few weeks, because N803 plus pembrolizumab, a really exciting study of an IL-15 agonist you'll hear about. So we're rolling. We've got more things in the work. You'll hear from Haas about our drug selection committee. So really happy that you're all taking the time to be with us today. And I see over 139 people online. Next slide. and I'm about to pass the baton. So now, who better to tell us about accomplishments than my friend, mentor, a former chair of the Lung Committee, David Gundera, one of the founders of LungMap. David, the floor is yours.
Hey, thank you, Roy. And thank you all for joining us. When we had, live meetings, of course, we did these in person, and I was always impressed how many people, even though they knew about LungMap, they would come because it was such a high priority. And so I'm going to very briefly talk about some of our accomplishments and also how the concept was developed, because it's being mimicked by people all over the world now. Next slide, please. So it started really, I think, with this report from the Institute of Medicine in 2010 that talked about how we need to reinvigorate the clinical trials program, the public clinical trials program, with 4 goals for modernization, and they gave 12 recommendations. But a lot of these were programmatic to improve the speed and the efficiency of trial development and activation, to incorporate more innovative science and also innovative trial designs, to improve prioritization, support, and completion of trials, and to incentivize participations not only by oncologists, but by patients as well. And if you look at these, and I read them out on purpose, they are the fiber of LungMap. If you can advance, please. So with this report then, there were 2 concurrent efforts that started at about the same time. One that I was part of was from the Thoracic Malignancy Steering Committee of the National Cancer Institute, and the chair of that effort was Fred Hirsch, who's with us today. And the other really came more from the Friends of Cancer Research Brookings Institute, and that was chaired by Roy Herbst. So of course, as you can see, as implied by the slide, we got these efforts together. Next slide. We met in Bethesda in February 2012, so almost 10 years ago. And you can see the attendees, of course, the NCI and our investigators, but also the FDA, a very strong proponent. Rick Pazdur was right on top of this. He led this effort as well as anybody else. We also had approximately 20 different companies represented who had drug development pipelines in the lung cancer space. And you can see the goals there were to put this together into a master protocol, and we issued a brief regarding the findings. Next slide. This is one of the slides that I initially prepared to say, what's the motivation, where do we start? And of course, we started in squamous cell lung cancer because this was the unmet need. As you can see from the adenocarcinoma, even then, in 2012, 2013, we already had a lot of targeted therapies either approved or in development. We wanted to have a master protocol where we could identify a therapeutic target and have a trial for that patient in at least 2/3 of the patient categories. You'll see to start, we accomplished that. And we felt that squamous cell lung cancer was really, at that point, and I would say still, an orphan group for targeted therapies. So we developed a master protocol. Next slide. And if you can advance once more. This tells a bit of the story, and you can see Ellen Siegel there, the leader of Friends of Cancer Research, who has been from day one a partner, but also an advocate for us at the highest levels. So we decided to start with squamous cell lung cancer. We spent a lot of time developing the infrastructure, and that was incredibly important because we found that our infrastructure is self-sustaining. You'll see in the next couple of slides, we had to make some major left turns or right turns because things were happening in the therapeutic space. We also wanted this to be separate from just an NCI program. We wanted it to be a public-private partnership. We accomplished that with the help of the foundation of the NIH. We developed a steering committee. We wanted a central lab. As you'll hear, we incorporated a partnership with Foundation Medicine, initially for tissue and now also for circulating tumor DNA in blood. And we wanted to have sub-studies that were independent of each other. You'll see this, it's incredibly important for a master protocol that one study can open, another one can close, and it doesn't affect the rest of the sub-studies. And then you see a nice picture of us together talking about a lot of these issues. Next slide. So this is a timeline. And again, I don't want to dwell on it, but you know, you look here from 2005 when the idea first came up to 2014, this was not business as usual. Every single partner here expedited the development of this program, and it came together with CTEP approval in June 2014. And then you can see all the participants on the right, as Roy said. Next slide. And this was our study design. It says original, but the theme is still the same. And that is, we would have an opportunity for a cancer patient, again, originally starting with squamous cell second line, that we would try to match a genomic marker and have a sub-study for them. But for those patients that didn't match, and this continues today, we would have non-match studies, the majority of which would be immunotherapy related. Next slide. So with that theme then, this shows how LungMap was launched in June 2014. So you can see we had 4 match studies and 1 non-match studies. I will say that every one of these targets, PI3 kinase, CDK4/6, FGFR, MET, all of these have drugs which are now approved. The majority in other tumor types, though, just pointing out that lung cancer remains a difficult target, even when a drug is clearly active in another setting. And you can see we had an initial non-match study with durvalumab. Of course, all of these are completed quite a while ago. Next slide. This again is a timeline for the first part of LungMap, which was called S1400. And although you can't perceive it so much from this slide, this is one of the pivot places, because all of a sudden, after we had launched LungMap and we were going along smoothly, immunotherapy was approved in the second line for squamous cell lung cancer, more than one drug. And so this made us rethink. You know, LungMap's been called a chameleon because we've had to change our face, but not our themes, multiple times. Next slide. And this brings us to the slide that Roy already showed. It shows this switch from S1400 to the name now LungMap. Everything in dark blue here is completed, 13 sub-studies. So yes, that is an accomplishment. As Roy said, we also incorporated some other themes as we learned along the way, like pre-screening, so that a patient getting their first line therapy could get the next generation sequencing done. And then when they progressed, we would know if they were eligible for one of our sub-studies. We have also modernized the biomarkers to now where we're using the foundation CDX. And also we now, for about 4 months, have been allowing patients who got this foundation test prior to enrolling in LungMap to use that information so that we don't waste tissue redoing the test. As Roy said, we've incorporated plasma circulating tumor DNA. Next slide. And that brings me to Mary, and I see somebody in the chat said, can we improve the audio? I'm not sure how I was coming through. I think it was fine initially, so.
I hear you fine where I am, David, so.
Okay, well, it may be that attendee, it's a problem at their end. Okay, so Mary, and I should say Mary from Copenhagen, because Dr. Redmond is joining us today from Copenhagen. So thank you, Mary, for taking part of your vacation time.
Absolutely. Yeah, so I just have a few updates to share about where we are with manuscripts to date. And David, that was a wonderful presentation. Thank you. So the next slide. This slide shows, as David was mentioning, we have our two series, the S1400 series and the lung map series. And this slide summarizes all the studies and ancillary trials that we conducted within the S1400 series and the papers that we have done within that S1400 series. I actually should say all of our sub-studies as part of S1400 have been published. And the ones highlighted in green are the ones that we published in the last year. And so I'm just really proud of this group to share that we have had great activity, and you can see on this slide where they're published, and please look them up if you're at all interested. And on the next slide, I share with you where we are with respect to the lung map series. We have completed 3 sub-studies within the lung map series. In that series, we have 1 non-matched trial, the S1800 trial, which is a randomized phase 2. We're waiting for the number of events to mature to be able to do the analysis for that. We're expecting to do that analysis this fourth quarter of the year, and hopefully we'll be able to submit those data for ASCO in 2022, and then the paper will follow shortly thereafter. S1900, which was looking at rucaparib, a PARP inhibitor in LOH, BRCA1 and BRCA2 patients. That was presented at ASCO 2021. The manuscript is in development, so you should expect to see that coming out sometime in early 2022. And then finally, S1900C, which looked at talazoparib combined with avelumab, that in STK11 positive patients. That study, the manuscript is in development as well. We're waiting for a little bit of the data to mature, but we're expecting to submit an abstract for ASCO 2022 and shortly thereafter submit a paper. So we've got a lot of productivity going on here. Next slide. That's it for me.
Thanks, Mary. David.
Yeah, so it's my great pleasure to discuss a little bit about our Translational Medicine Committee. If we could go to the next slide. Purpose of which is to bring together expertise in translational medicine to assist in biomarker selection and definitions and sample collection decisions for the sub-studies that are under development, to review proposals for prospective and retrospective translational research, and to assist in analysis and publication of sequencing and other correlative science data. Next slide, please. And yeah, I couldn't be more proud of this group. We have a wonderful array of lung map oncologists, statisticians, and folks involved in operations at SWOG and FNIH. We also come together with individuals at Foundation Medicine and at Nationwide at the Biobank to inform our best directions. So if you go to the next slide. Thought today, go over also some of the areas that we're particularly interested in, of course, the therapeutics, which, you know, some of the major categories these days being around immuno-oncology, targeted therapies, of course, DNA repair and anti-angiogenic therapy. But then also the other nuts and bolts that make a program like ours work, including how we handle best biospecimen banking and processing, look at pathology and clinical genomics. Next slide, please. So just to share a brief update on some of the resources that we've amassed to date, here is a summary of the sequencing data that we have from that first phase of lung map, which is S1400. This spans the dates between 2014 and 2019. Here we had an impressive 1,864 patients enrolled who have, with this first iteration, all stage 4 recurrent squamous cell lung cancer, as Roy mentioned. Here we have NGS data available for the vast majority of these patients, 1,672 of them. And this was done with a slightly older Foundation 1 T5 research platform that has a lot of overlap with the current CDX platform, but a few differences. T5 highlighted the exons and introns, about 313 cancer-related genes. So among these 1,672 patients, we identified a total of 36,000 some variants. And then you can see a table here at the bottom showing, the distribution of these types of variants, broken down by the type, including copy number alterations, rearrangements, and short variants. But then also about some of the significance, that's known about these. Many of the variants are known to have, an oncologic impact, but others have yet been, fully characterized. And you can see on the right here, a chart showing the number of variants that we've identified per subject, with a median approximately 20 variants. So and at this point, we have a description, of this data set, the prevalences of the different types of mutations, and have started to do some analyses on these. We go to the next slide. Just an example of which is this work that we presented as a mini oral World Lung a couple back. And manuscript is nearly ready for distribution. Here, it's one interesting finding. We did a mutual exclusivity analysis. So as I think many of you know, there are some genes whose alterations just don't tend to run with those of other genes because of something about the presence of one alteration that excludes the presence of another one. Perhaps they both hit the same pathway, and that multiple alterations in the same pathway could be deleterious to a cell, for example, or maybe you only really need one alteration in a pathway to cause, a biological effect. And so the cell doesn't see any pressure, to have those additional changes. There are many possible explanations, but here we've noted as one interesting phenomenon, mutual exclusivity between KEAP1 and NFE2L2, as well as a third gene, PARP4. It's been known for a while that KEAP1 and NFE2L2 are both parts of pathway that impacts responses to oxidative stress and treatment resistance. So an area of very strong interest in our group. But this PARP4 gene is really still yet under relatively little understanding. And so if we can understand how PARP4 is showing up as a mutually exclusive gene, we may learn a little bit about its function. And if it is indeed part of or impacts this oxidative stress pathway, may give us a new biomarker for study. Next slide, please. And then we've mentioned a bit about liquid biopsies, which I think will only become increasingly important over time. We have a number of studies with the recent series of lung map studies that have had plasma collected at multiple time points, good for these kind of ctDNA analyses. These include the S1900B and C studies and S1800A. There are slight differences in the time points, and our committee has been very interested in working toward, to the extent possible, standardizing some of these. But in general, one's looking at baseline, of course, as well as time points during the course of therapy and at progression as common themes. When we last cataloged this, we've had 537 samples, a very robust number available for tissue and plasma concordance studies, and we're working to get this number updated. Next slide, please. And here, Phil Mack and colleagues in a mini oral demonstrated some data showing the extent to which we are seeing concordance in the lung map study. Importantly, when you have a driver mutation, the type especially that we present in all cell lineages and rather than subsets of heterogeneous population, here we saw really good fidelity between plasma and tissue, but there was a sensitivity of about 83% of mutations seen in plasma that were found in tissue. So this is very reasonable and sufficient to suggest that the findings of on a ctDNA analysis could support enrollment onto lung map substudies, the positive finding meriting exclusion. But be careful that the specificity may not be there to use the lack of the finding of a ctDNA hit to exclude patients. I would not recommend making these the definitive by presence of their, by their absence. Next slide, please. We've also been very interested in cataloging, other TM resources that we have that we've developed along the way, including slides that FMI has been staining for PD-L1 and traditional hematoxylin and eosin. And here, when you add it all up, it amounts to quite a sizable resource. We have over 15 slide boxes with about 800 slides of these that could be useful for various analyses. Likewise, we have residual extracted DNA from the ctDNA submissions. This includes a very sizable 1,676 samples that we're currently getting assays for their residual mass quantities, so we know what we have to work with. And then some resources that are essentially fully renewable in that digital images, these data from these scanned H&E and PD-L1 slides could be made available to as many people as could find them useful. And so that's the type of resource we're also trying to cultivate. Next slide, please. And so, we want to make the use of all of this to the best way we can, because ultimately, we want to foster this goal to be able to, improve patient outcomes based on what I'm calling in general terms specified laboratory criteria, but basically to identify biomarkers that can help us identify in whom therapies would be the most likely to be successful. And so we want people to come with good ideas from our community, from study chairs, the academic researchers, and then the lung map collaborators, including Foundation Medicine and our pharmaceutical partners. Next slide. And we want to try to lower the cost of entry absolutely as much as possible. So we want to encourage these ideas. And so our committee has developed this letter of intent that we want to be really as painless as possible, just 1 to 2 pages, and we can get this out to anyone interested. And it's really just to request preliminary information on the proposed use of our bank specimens or data so that we can identify whether we have what you need, in terms of the data or biospecimens needed to power your work, and a little bit of information about it so that we can see how it might fit in with the lung map portfolio, and then work with you to develop your idea into a sound proposal. And so we'll make this available to everyone interested, and then I'd be more than happy to help look over this and then send people the right direction. Next slide, please. And this isn't meant to be overwhelming, but rather just to to highlight the fact that there are quite a number of stakeholders that are involved when it comes to translational medicine, including groups that want to really ensure that the science can succeed, including our statisticians, our operations group, our committee to look over the scientific merits of a proposal, and the other stakeholders, the folks who've worked hard on these sub-studies, the lung map leadership, so that we can really try to make a proposal as strong as possible before it undergoes review at CTEP and then ultimately gets CIRB approval as needed. We want to try to expedite and simplify this process as much as possible, but at the same time ensure that all the people who can help these proposals move forward has a chance to review these. But it'll all start with that letter of intent. Next slide, please. And just in terms of what we're really looking for, what would be a successful proposal, is really just to understand, how the science could lead to new scientific information, something that could ultimately impact patients. Would be lovely if what we can find may inform future clinical trials. We will want to understand how translational medicine fits within the platform, and then have a sense of what its priority is among other translational medicine ideas. We'd want to understand the background data and the justification for the proposed science. And importantly, we want to make sure that LungMap has the resources that you need in terms of the number of samples or quantities needed to make the science possible. And then this will all depend upon the timing at which translational medicine is proposed, whether as a study is getting going or retrospectively on the other end. And then we have processes in place for reviewing studies at any of these points in time. And some questions that may come up along the way include, for example, if results could be potentially pertinent, how they might be provided to the treating investigator or patient. I think that may be most of the slides I have. Next slide, please. All right, because I think we'll hold any questions to the end. Thank you.
Great, thanks so much, David. So I'm Jyoti Patel, I'm the Vice Chair of LungMap, and I'm going to be presenting on behalf of Dr. Borgai, who unfortunately couldn't be here today, but has been the chair of the Drug Selection Committee for several months now and has really done an extraordinary job. Next slide, please. So first I'd like to just reiterate what the drug selection process is. I mean, this is, again, ideas in LungMap, I think, are so closely vetted with informal advisory boards working with pharma as well as advocates and other stakeholders to really develop the best trials. So when we think about bringing a new compound or drug to LungMap, we really are casting a very, very wide net. We want suggestions for sub-studies to come certainly from LungMap PIs, but we're also looking at outside investigators, junior investigators within cooperative groups, and from pharma itself. Usually when we have an initial idea, there's a small internal discussion with a smaller group of PIs. We have informal presentations. These are, again, highly vetted, a lot of candid feedback to really make the proposal as strong as we possibly can. After those discussions, the concept then goes to the full drug selection committee. And that application is a little bit more full. It includes preclinical PK and PD data, prior experience and safety, all of the information that one would need for a new compound, essentially. And that presentation is again led by the investigator and pharma, and it's a closed committee discussion. There's a formal voting process. We think about target appropriateness to non-small cell lung cancer, really where do we have the best data to move combinations forward. We then go back to our industry partners and pharma and let them know what the results are. And then once we've accepted a concept, it goes to SWOG operations and FINH with a sort of this kickoff call and regular teleconferences to really keep this timeline moving and to ensure timely development of a concept. Next slide, please. So the steps for adding a new sub-study, and again, this is casting as broad a net as we can. First, it comes to the drug selection committee, and then we really look at concept development. So timely development for biomarker selection and definitions. We have parallel, that is in parallel with CTEP concept review. You then go on to protocol development, and this is really where the SWOG support is essential with rapid teleconferences, statistical review, budgets and contracts are all going in parallel. Finally, it goes to CTEP and CIRB review and then activation. And we want to make this timeline obviously as tight as possible. Next slide, please. So here is sort of the high level overview of sub-studies. So the drug selection and concept development, again, this is really where the meat of the work comes, in which we're trying to develop a concept which is timely, is sort of looking forward to initiation in maybe 6 months and will remain relevant for multiple stakeholders. And you can see that all of these things are happening in parallel. And we've been able to really cut down on times recently. So really exciting how different committees have come together. Next slide. Concepts that are currently in development. We have a host of concepts right now that are making their way through, and this reflects certainly biomarker-driven studies in rare populations, as well as concepts in the IO refractory population. So the first concept that is going back to DSC, the PIs are Sarah Goldberg and Ross Kamage, and it's osimertinib plus capmatinib with or without ramucirumab. A similar ramucirumab trial with tepotinib is going on and is moving forward, and Paul Paik and Shunming Lee are chairs. It's a TIGIT concept, Infinity Pharma, that myself and Mohammed Furqan are working on, which we hope will be a triple arm study. Regeneron, we're looking at the bispecific MET antibody. Jimmy Ruiz and Becca Heist are leading that. We have the ATR inhibitor with durvalumab. Next slide. And then other concepts that are still a little bit earlier in discussion, but we're moving along. So we're looking at TROP2 ADCs, AXL inhibitors, and EGFR MET bispecific antibodies. Again, some of these are approved, but we're looking at combination trials. We're looking at refractory populations. And so certainly ones in which full biomarker and translational science that David discussed would be of significant interest. Okay, next slide. And a couple of things that we know are gaps and really unmet needs for patients. So we've been in conversations with Kinnate and Amgen and Genmab. We're looking for pan-KRAS inhibitors as well as other ADCs. So certainly, you know, this is a really robust net, but I encourage any of you who have ideas to come to us. We're anxious to really get the best trials to patients all over the country. Next slide. So with that, I'm going to hand over to Dr. Pata, who's going to talk about a study that is ongoing and is a very high priority right now for LungMap.
Thanks so much, Dr. Patel. So I'll be discussing S1900E. This is a phase 2 study of sotorasib in patients with previously treated KRAS G12C mutated non-squamous non-small cell lung cancer. I'm the overall chair of the study, and Dr. David Gerber is the co-chair from UT Southwestern. Next slide. So what do we know about sotorasib in this patient population? So these are the most up-to-date results from the phase 2 CodeBreak 100 study. This included 124 patients treated at the 960 milligram daily dose. And here at this data point, we have a median follow-up at 15.3 months. So in this population, the objective response rate is 37.1%, disease control rate 80.6%, median duration of response of 11.1 months. If we look at the PFS and OS, there's 6.8 months and 12.5 months respectively. Next slide. So are there particular biomarkers for sotorasib? We know that co-mutations in KRAS mutated lung cancer have had some values as it relates to prognosis and also potentially predictive value as it relates to some therapies such as immunotherapy. So on the left, as part of the CodeBreak 100 study, they looked at co-mutations of P53, STK11, and KEAP1. And what we can see is that the objective response rate in both the P53 and STK11 groups looked very similar to the overall population. But in the KEAP1 co-mutation group, they tended to have lower probability of response. But what's important to note is sometimes these co-mutations can co-occur with one another. So that's when we get slightly different results on the right in the STK11 mutation cohort. So if you only look at those STK11 mutation tumors in conjunction with KEAP1 wild type, you actually see a different signal emerge, that the objective response rate may be slightly better than the overall cohort and other subgroups, and that the median progression for survival and median overall survival also look quite good, although the sample size is relatively small for these sub-analyses. Next slide. So the next thing I'd like to point out is this is, of course, a very interesting analysis that was done in a retrospective fashion as part of the CodeBreak 100 study. But if we look across the co-mutation subsets and we look at the 95% confidence intervals around the point estimates of efficacy, some of them are quite wide. And I've made it easier for you to see, hopefully, that those are noted in red. So as an example, if you look at the STK11 mutation cohort with KEAP1 wild type, which is very much an interest to all of us, the confidence interval around that point estimate of 50% ranges anywhere from 28 to 72. So I think we still have more work to do in terms of knowing how well these drugs work in each of these different co-mutation subsets. Next slide. So there was additional data that was presented at World Lung this year, also looking at molecular features as it relates to different endpoint with sotorasib of early progressors versus late progressors using the 3-month threshold. And again, we can see that KEAP1 co-mutations are really associated with early progressive disease. STK11 didn't have a particular association with any of these categories. Well, there were a couple of different pathways where alterations were noted, such as in the cell cycle and Wnt pathway, that were associated with late progression. So we have much to learn about the genomic contexture of KRAS mutated lung cancer, including KRAS G12C mutated lung cancer. Next slide. So this is a S1900E schema, and we know that sotorasib has been approved, but I'm going to take you through the schema and hopefully relate to you why this study remains important. So the S1900E sub-study has similar eligibility to CodeBreak 100. So these are patients with recurrent or metastatic disease, KRAS G12C mutations, and non-squamous non-small cell lung cancer. They also have to have been previously treated with at least one line of treatment, whether it's immunotherapy, chemotherapy, or a combination chemo-immunotherapy, and no evidence of uncontrolled brain mets. So the first important part of this study is that for the biomarker assignments, this is an upfront biomarker definition, and there are 3 cohorts. And first, the P53 mutation cohort. Part of that cohort definition includes wild type of STK11 and all of the KEAP1 axis mutations. This is also true for the STK11 mutation cohort, where there also has to be evidence for wild type P53 and all of the KEAP1 axis mutations. And because we know, based on the lung map data itself, that these co-mutations can co-occur in 30% of P53 and up to 80% of STK11, we wanted to ensure that patients can still enroll. So there's that final cohort that allows those patients with dual or even triple co-mutations to enroll. So once the biomarker definition has been met and eligibility has been met, the patients receive standard dosing sotorasib, 960 milligrams daily. The treatment period is relatively reasonable. They're seen with in clinic and labs with every cycle, and imaging is conducted every 2 cycles. And one of the other important parts of S1900E is that all patients at the time of progression will have a liquid biopsy. So we'll be able to very much interrogate not only primary resistance of this drug, but also acquired resistance of this drug. Now, the primary endpoint is objective response rate. Mary Redmond, the statistician for this study, has estimated that the 95% point estimates, or the 95% confidence interval for the point estimates should be more robust than what we've seen in the exploratory analysis of CodeBreak 100. And if we look at all the different cohorts, the sample size totals 105. So we're adding information from another 105 patients that were treated with sotorasib to the CodeBreak 100 study, where 126 patients were included. Next slide. So right now we do have an interim analyses built into each of the cohorts. This was of course done before we had the FDA approval. We anticipated that based on the estimated percentage of the study population, that it would be relatively faster to accrue to the P53 cohort and the all others cohort. But if you look at our accrual numbers, actually, as of October 7th, we're seeing accrual even quite well across the STK11 cohort. So in P53, we have 11 patients, STK11 cohort, 7 patients, and then the all others cohort, we have 13. So on our wish list is also to expand that STK11 cohort so we can learn as much as possible. Next slide. So this is my final slide. So these are some lessons and tips as S1900E has now been open. So I think one of the most important things, especially since sotorasib is improved, is to screen patients early on lung map, specifically while they're on frontline treatment. And this not only includes potentially submitting archival tissue, but also if they've already have foundation medicine testing, submitting that report. And this allows patients to move on more quickly to sotorasib at progression if they are eligible. The second thing that I think was also mentioned earlier in this webinar was that cooperative groups are more likely to enroll diverse population because they are also include a variety of sites within the community in addition to our academic centers. So if we look at race ethnicity breakdown within Codebreak 100, there were 82% of white patients, Black patients at 1.6%, Asian patients at 15%. So there was a proportion of patients of Asian race who were enrolled. However, if we look at, all the patients who have screened on the LungMap screening protocol, and these are the latest numbers, the race breakdown includes 86% white, 9% Black, and 2% Asian. So really is the hope that we will have an increasingly diverse population that will add to what we know about sotorasib in a diverse population. All right, in final, we have a protocol amendment wish list because we of course want to make things as easy for all the sites and investigators and patients, especially with the approval. So we want to simplify eligibility, shorter washouts from prior treatment, simplify drug-drug interaction language, liberalize laboratory criteria. In terms of the statistical design, I previously mentioned we would like to very much expand the STK11 cohort because there's a lot to learn there. And then finally, since the drug is approved for all patients with KRAS G12C, we would like to remove the interim analyses. So I thank you for your time and thanks to the chairs for the opportunity. Oh, next slide, I can introduce our next speaker. Okay, so next we have Hatham Hussain from UCSD discussing the S1800D substudy.
Hello, everyone, and it really is a pleasure to be here. Thank you to the SWOG leadership on behalf of John Rangel and myself to present S1800D, which is a phase 2/3 study of the molecule N803 plus pembrolizumab versus standard of care in patients with stage 4 recurrent non-small cell lung cancer previously treated with a PD-1 or anti-PD-L1 antibody. Much of the work that I'm going to be presenting is actually been spearheaded by John Rangel, who is at the Medical University of South Carolina, who's really paved the way by leading the phase 1 study of this molecule N803 in defining some of the translational endpoints of the study. Next slide. So we're really excited about this study, partly because it provides an opportunity for patients in an unmatched category. And again, this is looking at the molecule N803 plus pembrolizumab to overcome PD-1 resistance. There's enrollment into 2 categories of patients, those that have primary resistance to PD-1 therapy and those that have acquired resistance to PD-1 therapy. The primary endpoint is overall survival in both of the cohorts. And the design is for the primary resistance category, a randomized phase 2. And the acquired resistance, which has a larger sample size, a phase 2 and 3 design. Next slide. So as I mentioned, John Rangel at the Medical University of South Carolina has led a phase 1b analysis of this molecule, which I'll go into a little bit of depth around. And this was published in Lancet Oncology. Again, looking at this molecule N803, or otherwise known as ALT803, which is an IL-15 superagonist. And the original study was done in combination with nivolumab. And we're going to be extending these data sets into additional PD-1 molecules, including pembrolizumab in this study. Next slide. So there's more and more literature and work integrating novel molecules targeting cytokines in addition to PD-1 antibodies. Again, one of the cytokines that we all have had familiarity with is IL-2, which approved in certain tumor types has higher toxicity than some of the other cytokines that have also shown preclinical efficacy. IL-2 is a family member with IL-15, and both actually have the effect to recruit and activate CD8 T cells and also NK cells. And preclinical work has shown that in combination with PD-1, this can be synergistic and actually also have effects to improve outcomes in the stage of acquired resistance. So the way that IL-15 works differentially compared to IL-2 is that it can expand the CD8 and NK cells, but spare CD4. And in sparing CD4, that has the effect to reduce some of the toxicity that has previously been seen with IL-2. Next slide. So here is a patient from Dr. Rangel's practice in which the patient actually was a 65-year-old female who had KRAS G12C as well as PD-L1 overexpression at 70% and had gotten carboplatin, pemetrexed, and pembrolizumab as first-line therapy. And the patient did have response on that therapy, but progression only 5 months later. So the patient was enrolled in on his study of the phase 1 with this molecule and had actually really brisk and deep response early on the first scan, and that persisted. And in fact, actually, the patient is now 4 years out of this therapy, still with ongoing response. Next slide. So here's a waterfall plot, which capitulates the 21 patients in his phase 1 study. That study was divided between those patients that were treatment refractory, meaning had progression within 3 months of therapy initiation, as well as those that had initial response to first-line therapy and then progression, relapse patients, and those were defined as relapse within or after 3 months of therapy. Particularly, there were 3 patients that had PRs in this relapse group, and many of those patients had ongoing responses. The PFS in that phase 1 study was 9.4 months, and the overall survival was 17.4 months, which was quite impressive. Also importantly in that study, there was stratification based on PD-L1. And in the cohort of PD-L1 negative patients, which were about 11 patients, the response rate was 30%, with a disease control rate of 70%. Next slide. So here are some of the toxicities that can be seen with this molecule. And this is actually a really great segue into saying that both John and myself are very available to take any questions throughout the trial on this, and really good infrastructure to work with the study coordinators at each of the sites to help in any ways. But some of the basic points actually were that the toxicities were quite quite manageable. In fact, the injection site reaction rate was seen, but primarily less than grade 3 events. Some of the other toxicities that can be seen were fatigue, some flu-like symptoms, some rare circumstances of hypotension, fever, nausea, anorexia, and chills. And again, this is an area that both John and myself will work very closely with all involved. Next slide. So here is a slide which shows the study design. As I mentioned, patients must have received prior PD-1 therapy and disease progression. Again, there are 2 cohorts. One is the primary resistance cohort, for which there's about 130 patients included in that cohort. And again, the randomization in that cohort is between N803 plus pembrolizumab versus standard of care therapy. And the standard of care therapy could be docetaxel ramucirumab, docetaxel, gemcitabine, or pemetrexed. And then there's a larger cohort of acquired resistance. And again, that's defined as greater than 12 PD on or after therapy, PD on prior therapy of greater than 12 weeks. And the randomization in that cohort is N803 plus pembrolizumab versus standard of care with the same agents being evaluated in the standard of care. Next slide. So just wanted to thank you all, really excited on behalf of John Rangel and myself, looking forward to working with everyone, and happy to field any questions throughout the trial. So we're now gonna have a discussion about the Site Coordinators Committee led by Stephanie Smith. Thank you.
Hi, good morning, everybody, or afternoon, depending on where you are. Can you guys hear me okay? Are you able to hear me okay?
Yes, Stephanie, very well.
Okay, just wanted to be sure. I am Stephanie Smith. I am the current chair of the Lung Map Site Coordinators Committee, and I'm here to talk about what we do. You can go ahead and go to the next slide. So the Site Coordinators Committee's mission is to represent study site staff among nursing, CRA, data management, and regulatory levels by providing feedback to and from the study leadership to enhance accrual and improve study management. We were developed about 5 years ago now, and we continue to be a prominent part of the lung map study. You can go to the next slide. So our members have come from a broad range of sites. We have members, we've had past and current members from small town rural cancer centers to large NCI designated cancer centers, which is ideal in a committee like this. So by doing that, we can provide real world boots on the ground type of input into lung map. Some of the ways that we do this is by reviewing and recommending accrual materials and strategies. We've provided content for the Lung Map newsletter and regularly participate in these Lung Map update meetings. We also present at OISHI. We've presented there in the past, talked about Lung Map, talked about our committee. And we're all champions of promoting this study and encouraging accrual within our NCOR or individual institutions. So we are basically Lung Map cheerleaders at our institutions, if you will. You can go to the next slide. Our members also assist with training tools. We review lung map protocols. Occasionally, we're asked to give feedback regarding the availability of supplies or availability of certain specimen tubes at our institutions. In summary, we are essentially lung map site representatives who provide lung map leadership, true insight into this trial from a coordinator's perspective, which overall makes this a better trial. So the Lung Map Site Coordinators Committee needs you. If all of that I just mentioned interests you, if it sounds something you'd be interested in, we would love to have you join our committee. We are currently filling multiple vacancies within our committee, and nobody knows the nuances of coordinating this trial better than you. We're seeking all physician site representatives who work on Lung Map. We need input from every type of institution, so don't be afraid to apply. Nurses, CRAs, data managers, regulatory staff from across the NCTN are all welcome. Interested site representatives can fill out an application through that SurveyMonkey link that you see on your slide, or you can email the Site Coordinators Committee and we'll be happy to reply to you with the link. The next slide. If you have any questions, any feedback, you want to bounce anything off the Site Coordinators Committee, feel free to reach out. Our email address is on the next slide. There should be one more slide after that with our email address. You can reach out to us. And finally, these are a couple of pictures of past and present Site Coordinator Committee members back when we met in person. Hopefully we'll be meeting in Seattle, taking a new picture in the spring, and maybe your face will be in the photo. Thank you.
Next slide, please.
Thanks a lot. I was just taking a screenshot so we could put that up on the Twitter. The engagement is just so important, and we want to hear everyone's voice. And one of the best things that's happened to Lung Map in the last year is getting Jyoti Patel so involved as the vice chair. And Jyoti is going to take us through the future of Lung Map, and then we'll have a lot of time. We have a lot of time for discussion today. Jyoti.
Great, thanks so much, Roy. And Stephanie, thank you. That picture on the river is certainly a nice one of Chicago, so we hope to see you here again. So we're going to wrap up, and I really want this to be sort of interactive as we think about how LungMap has evolved, really, the journey that David described earlier in the session. It's important for us to make sure we're recalibrating and keeping our eyes on the prize. Next slide, please. So as has been stated, and you saw this slide at the beginning, this is truly a partnership, and that's what makes this, I think, a unique part of our national trial system in the United States. It's truly a public-private partnership in which we are trying to get the best science from investigators, from industry, and then really thinking about our accountability to our stakeholders, which are our patient advocates, and our patients in really transforming how we do clinical trials and bringing trials to patients. Next slide. But a lot has changed, and I put this sort of Ted Lasso-ish piece in. The goalposts keep on changing, right? So certainly when this was first, when Fred and David and Roy really started this conversation in earnest, next-gen sequencing was truly a unicorn. And now we know that it's widely adopted, particularly for non-small cell lung cancer. We've seen, unfortunately, data recently at international conferences that we're still woefully inadequate. There are a lot of patients that aren't getting testing, but we have good level 1 evidence to suggest everyone should with advanced disease. We also, since this time now, have immunotherapy as truly a standard of care for most patients with lung cancer in the frontline setting or in combination with chemotherapy. During this time, we've made tremendous strides in blood-based testing in terms of sensitivity, data about concordance, as well as cost savings by doing upfront blood-based testing. And now many of us think that it's a routine in our care to also assess for mechanisms of resistance. I think the biggest thing that we are facing in lung cancer is great because the world has changed so quickly, but it's really tough because we have really ambiguous historical standards. The field has evolved. So we can't say what a patient, how a patient did with a particular mutation in an era before immunotherapy when that patient might have been exposed to immunotherapy or treated successfully with immunotherapy in the frontline setting. So certainly I think this idea that we have to remain limber is an important one as we think about what we do with our next steps. So next slide. In response to the fact that that now next generation sequencing, genetic sequencing is so widespread and widely adopted, for a long time, that was sort of our value add for patients. And now many of our patients are having sequencing either within their institutions. If you want to go back a slide, please. There you go. Or I think go back. If you could go back. Oh, I'm sorry. Go up one more. I'm a little messed up now. So there's been a strong call for letting us use existing testing in terms of tissue stewardship as well as redundant analysis. And so for the past year, we've now been able to update eligibility criteria to allow outside Foundation 1 CDX testing. And so that's been going on for the past year. Essentially, patients who have pre-existing Foundation testing can submit their CDX results. Once that's done, and it's been done since 2019 on solid tumor, then we can reanalyze the commercial test results, get them into the LungMap system. It's usually about a 2-week turnaround, and this has not been used significantly, I think, because we have had fewer ongoing studies right now, but we're seeing a little bit more of this, so keep this in mind. Next slide. So this is sort of the information that you need if you're submitting commercial Foundation 1 testing. Next slide. So that's, again, been for these past several months and hopefully will lessen the burden Many have asked us to sort of increase the number of testing platforms that we use, and we understand that there are many other vendors that are providing really good information. We recently did a site survey, and it sounds like most people are still using Foundation Medicine, but we're continuing to work to find if these other platforms would make sense for us to include. So again, responding to changing times. Next slide. I think the big piece of really, the question that David Cazono talks a lot about is we have so much translational medicine. We have a huge wealth of information about mutations and co-mutations. And so really, how can we leverage that information to get the best therapies? So sort of our major buckets right now of treatment are, I think, truly in identifying and overcoming mechanisms of immune resistance. And as Dr. Hussein presented the IL-15 study, you can see that we're really looking at primary and acquired resistance. We're looking to identify rational strategies who have also, for patients with targeted therapies. And so that I think is exemplified by the capmatinib, osimertinib, and ramucirumab trial that we hope to get moving under the leadership of Drs. Goldberg and Kamich. These are small patient populations, and so having a network as robust as LungMap is certainly important. What's really nice, though, again, is this comprehensive biomarker analysis that we have from blood and tissue that will provide insights, and that I think is exemplified by the work that Dr. Pata is doing. And very importantly, we really need to work to develop lung cancer-focused clinical researchers, and this provides us an opportunity to do that and really find early ideas and give infrastructure and options to investigators who have great ideas. Next slide. So we sort of see it as perhaps not just a circle, but really a path forward, right? There's collaboration from multiple stakeholders. We have a generation of lung cancer clinical researchers that are ready to take the reins. And the focus of LungMap remains patient-focused and patient-centric. So innovative trials in the community at a patient's behest. So again, we remain really optimistic about the trials that are coming through and the ongoing trials, and encourage you to continue to support LungMap. So with that, I'd like to open for any questions to all of the panelists. You can use the chat button or you can raise your hand.
Great,
Roy,
thanks.
any other comments?
No, that was wonderful. Why don't we take down the slides and put all of our faces up? And I'm going to encourage people to show their faces for a couple of reasons. One, so we can get to know each other again. And two, David Gundera, I know, wants to get a screenshot. I just know him after all these years. So put your faces on and let's if someone can help me and Jyothi to find the hands. You can raise your hand using the hand function. You can just start to talk. But we've got 150 people on. I'd love some comments. What do some of the people, the member of the committee, feel about the new trial coming forward that you heard about it? Anyone have any thoughts on that? How that's going to fare in the community? Hatem, just reiterate, when do you expect we'll have first patient in?
So the study will be activated in November. So hopefully, very quickly. This is actually a nice study because we all have many patients who are progressing on PD-1. And unfortunately, there are always, may not be great options for people in standard care settings. So kind of really moving the needle to kind of have this there, I think is, hopefully we can accrue quite well.
Excellent, thanks. And we haven't heard from Karen Kelly, our chairwoman, she had a whole great series of meetings so far at SWOG. Karen, your thoughts as we move into this next year of LungMap?
So I just want to first of all say congratulations to everyone on the LungMap team. I thought the presentations today were just outstanding. And I think it really does provide continued enthusiasm for developing new agents and testing them in this patient population. We still have a long ways to go, and this is a great mechanism to do that. Yes, we have some, infrastructure work to do, and we all acknowledge that, and we're doing that. But I just want to say kudos to everybody, because you see all the people on this call. Everybody is interested, excited about LungMap. Hadam, I think this trial is going to accrue like gangbusters. Should accrue incredibly fast. And I personally am so excited about it, and all the upcoming studies. I think it's great to have Jody on, and Haas and David Kuzno. They have done an outstanding job now as being integrated into the LungMap team. And so I think that having all of the groups in this has really been fantastic. And I just look forward to really more exciting trials to come, and to the publications, and to just really mining the data. I just think that this is a fantastic platform. And just again, congratulations to everyone.
Right. Some questions are coming in. I've got one here. It's to the panel, and it says, we love LungMap at our site, but we're a little frustrated. There haven't been that many open sub-studies, and that's limiting. And I guess to the group, how can we speed that up? Jofi, do you want to start, and then maybe some of the others can chime in?
Sure. I think that reflects sort of the pause that many of us have had sort of in the post-IO era, and just now we're coming up to speed with new concepts. So as Karen and Hatim mentioned, this upcoming trial in immunoresistance is really exciting and a huge unmet need, and hopefully that will move quickly. And it's probably about a month away from activation, and so we anticipate that will be that should move quickly, and hopefully will be activated at your sites. And then certainly, the sotorasib study has been a little bit tough, because approval came right as we were ready to activate it. But actually the timeline for activation was short, and it was really speedy. Again, for patients, there's a lot to learn still about this drug. Many drugs now are approved on cohorts of even 80 patients, right? And so for us to really understand and better understand mechanisms of resistance and great responses, it's incumbent on us to, I think, capture this data prospectively. And so, I hope some of the trials that we're doing really reflect that. Trials particularly in the exon 20 space that we've been talking about may further really give us some knowledge to look at broader patient populations, more diverse patient populations, and have a better handle on therapies.
David has his hand up, Jofi.
Yep, go ahead. I'm sorry, David.
So I just wanted to reiterate and expand a little bit, Jody. So I think, some of our, most ardent participants in LungMap don't realize that in a way we're victims of our own success. As you saw, we've already completed 13 sub-studies. And most recently, several of these sub-studies have accrued far exceeding expectations. So we're a wonderful machine. And I'll just quote you, Roy. So Roy, I was on a meeting with Roy recently, and he said the store is open. So the store, of course, here is LungMap. And he said, we need more products on the shelf. So the products, of course, are the sub-studies. So I thought it was a fantastic quote. So for any of you who are from industry, we want your best ideas. You saw in the slides that I counted 14 new proposals which are in development. Not all of these will make it to activation, but if even half of them make it, there will be enough products on the shelf where we can go back to our original concept of having a high hit rate for patients who enter the study.
Thanks, David. I have another question for David Kozno. David, this one is from a young investigator, I won't say who, and she says, my chief wants me to write an R01, and I need a validation set. How would I get access to work with you and all the samples that you have for LungMap to partner and collaborate?
Yeah, best way I think would be just send me an email. I'll put it in the chat right now. But happy to help.
And a similar vein, Mary, there are so many, the statistical group here is just amazing, the whole operations. There's so many questions, but of course, people have to come with hypotheses and want to work interactively. Mary, what's the special sauce here? How do we make more of that happen? Mary's on mute.
Sorry, I have a lot of Danish speaking in the background.
Well, Fred will translate.
I can help you, Mary.
Well, yeah. So how do we make it happen? We have a whole process. And I mean, for one thing, if people want to access the clinical data, the studies, I mean, we post a lot of the data up on, I mean, the phase 1, phase 3 trials are posted at the NCTN data archives. People can look at the SWOG website to see how we can share data. So there are policies that are posted, but they can email our team. So our goal is to share data once that we've published the trials, and we can either collaborate on the work or we can share the data. There's a different process depending on what needs to be done, but any data that we have should be used for further purposes.
Thanks, Marion. Stephanie, here's a question for you. How do I become a member of your committee and get more involved? It looks great. Stephanie Smith.
Hi, thank you. Sorry, I was trying to find my unmute. There is an application process. You'll complete the Survey Monkey that we can send it to you if you'll email SEC, our alias there at crab.org. We'll send you out the application. We'll put that in the chat for you.
Okay, Suki, here's a question for you. They were impressed by the fact that you have, the trial that resulted in the approval of Amgen 510 had almost no minority patients. Actually is horrible. The lung map seems to be doing better. Any thoughts on that? And I know you mentioned it a little.
Yeah, I think there's been data presented, and I forget, someone said it earlier in the session that cooperative clinical trial groups do a better job of accessing diverse patient populations or going to the sites where these patients are, because it's a mix not only of academic centers, but also community centers. So, of the patients in S900E, right now, 27 have been registered and are eligible. So we'll have to see, in the end, what the proportion is of race and ethnicity and how much more diverse the population may be compared to Codebreak 100. And I know from Amgen's perspective, this is also a very important part of working with LungMap on this study, not only for the co-mutation subsets to learn as much as we can, but also for this particular point you brought up about enhancing diversity, patient diversity in accruals.
Okay,
I could just add, Roy, that we have strong VA participation at several sites, and of course the VA has our ex-military personnel, and is an area of increased diversity in terms of institutions. So I think that helps our minority accrual as well. And it also gives those veterans, access to these new therapies.
Thanks, David. Judy, this is all about patients and access and, raising the bar on cancer. And I know you have your finger on the pulse. Any thoughts? And perhaps after you talk, we might have any questions. We're last call for questions in the chat, please. Judy?
Sure. Well, I put in the chat just in case I couldn't get to it, but I'm really excited to see that there are 6 new concepts being developed and 3 being talked about, and the potential of liquid biopsy. I've always thought that would be wonderful if it could be more reliable with the specificity and concordance as David presented in his slide. I'd like to see that happen in the future, and I know patients would really be happy with that. Also, the amendment that went through to use the foundation CDX tumor results from before, that's so huge in my opinion, and I'm sure patients would be happy about that. Other than that, I think LungMap has a lot of potential, even though there are only 2 right now, but the 2 that are out there seem very, very exciting that patients would want to participate in with those rare mutations. So I'm really excited about it. And I really look forward to continuing to work as a part of this team to watch what happens as we move forward.
Thanks, Judy. Anyone else from the panel want to say anything before we end? And then I'll give the last word to Jyothi. We have no more questions left in the question area. We've had some good questions. I'm looking at the group here. Fred, how can I not ask you a question? Fred, has the bumper sticker. Tissue is the issue. So, Fred, what would you do? So if you had access to the specimens, what are some burning questions we could do and answer with the lung map specimens?
Now, first of all, congratulations to everyone. As David Gundera mentioned for me, this was our child, which has now become a teenager, and it has been wonderful to see the development. What we can do with the tissue in the future, I think still it is a complementary thing with tissue and liquid biopsy. We need to pursue both of them. What I think is probably the future is, of course, combination of therapies. But when we are talking combination of therapies, I'm very much an advocate of looking into combination of biomarkers. And I think that is our challenges in the future. We have had a tendency to focus on just one single biomarker. I think we need to focus more on combined biomarkers, panel of biomarkers. And I will re-emphasize, Roy, what you said. LungMap is a wonderful opportunity to do validation studies in grant proposals. So I will certainly encourage people to look into that as well.
Right.
Congratulations with the development.
Okay, we're at the last minute, but I'm going to say one word and then turn over to Jyothi. And what I want to say is that, well, David says we are a store. We're actually more like Amazon because we work with all companies. And one thing that I feel very strongly about is that LungMap now really is all the cooperative groups getting together. I see Takwa El Hosseinon and Stacy Adam from the FNIH. We really are all working together. In fact, Jyothi's, we're happy to have Jyothi at our SWOG meeting, but Jyothi actually has a main allegiance at a different meeting, but it doesn't matter because it's about the patient. And, we're talking to the NCI later today. And one of the things that, I feel so strongly about is it's about the community of patients with lung cancer and how we can all as a team find the best trials to get the best drugs to the patient at the right time. So with that, Jyothi, the final words are yours. We're right on time. And it's just wonderful having worked with you and the entire group of leaders this year. Thank you for all you've done.
Thank you so much. I mean, this is such a group effort and a fantastic team. So it's been really just outstanding to see all of the groups get together and work with the FNIH and the NCI to really, again, bring our industry partners shoulder to shoulder to focus on what patients need. And so thanks for this tremendous group effort, for everyone's leadership. Stay in communication often. If you have questions, reach out. But we certainly have our work cut out for us, but it's an exciting time. And so I think many hands make swift work, so we'll get there. So thanks so much for your time, everyone. Please reach out with any questions or suggestions.
Okay.
We'll see you soon.
See you all soon. Okay, thanks everyone. Go and roll.